Hi, thank you for joining us today in CCeMMP seminar series. I would like to begin by acknowledging the Wurundjeri people of Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of various lands on which you all are joining me from today. I pay my respect to their elders present, elders past, present, and emerging, and to the Aboriginal elders of the other community. I am Bhavika, a Central PhD student at Monash Node. Before starting the seminar, I would like to remind that the seminar will be recorded and will be available on CCeMMP website. I would also like to mention that the presentation will be 45 minutes followed by 15 minutes Q&A session. So please save the question to the end of the seminar. You can either raise your hand or pop the question in the chat box, and I can read them for you in the end. Please stay muted throughout the presentation. Today, we are very excited to have Professor Isabelle Rouiller. Professor Isabelle is a Deputy Director and University of Melbourne Node Leader of CCeMMP. She is a pioneer in the field of cryo-electron microscopy. She has applied this technique for over 20 years to study macromolecular complexes critical to health and disease. She obtained her PhD in 1998 from Institute of Animal Health in UK, where she worked on assembly of African swine fever virus. She pursued a couple of postdoctoral training in single particle cryo-EM and electron tomography at Scripps Research Institute and Burnham Institute in San Diego, USA. She then moved on to establish Canada's first cryo-EM facility while building her independent research program at McGill University. In 2017, she relocated to the University of Melbourne, where she's the head of cryo-electron microscopy laboratory for studies of molecular machine, focusing on understanding protein function in health and disease. In her lab, she explores the molecular mechanism of proteins involved in membrane fusion, including HIV, ENV glycoprotein, and OPA1, DRP1 GTPases, the potential ion channel TACAN, TACAN, and AAA ATPases, P97, also known as VCP. Today's seminar will be focusing on her groundbreaking work on VCP. So welcome, Isabelle. Over to you.
Excellent. So let me share my screen. Thank you very much, Bhavika, for this kind presentation and for your willingness to share this seminar. And thank you, everyone, for joining. I assume most of you are not at Lawn Protein, not on the beach, but in the lab, working hard. So thank you very much for sharing your time and spending time with me. So today I will talk about VCP or P97, and I will talk about, my talk will be organized in 3 sections. I will first start with a short introduction on VCP. Then I will present 3 short stories, one on the molecular mechanisms of VCP disease, focusing on our work that has shown that conformational impact of disease-associated variant. The second story I will show you is I will present an image analysis method to characterize the conformational heterogeneity from cryo-EM data, and I will demonstrate the application of this method to the pseudo-symmetric particle of VCP. Then at the end, I will show you some of our recent work on the mycobacterial DNA-Ku Non-Homologous End Joining synapse complex. And this is a very compact, unstable complex that is disassembled by mycobacteria homolog of VCP. And then I will finalize my talk with a few concluding remarks. If you have any questions during the talk, feel free to pop them on the chat as they come to your mind, so that you don't forget about them, and we can address them at the end. Okay, so VCP or P97 was first discovered in 1990. And it was discovered as a ring-shaped particles about 12.5 nanometers diameters. And then the yeast homologue at this time was reported 5 years later as to be important for the fusion of ER membrane in yeast. Then, during my postdoc, I started working on this complex, reporting that P97, or also called VCP, as we will see later, is undergoing conformational changes during the ATPase cycle. So first we showed using cryo-EM and 2D averages that we had an hexameric complex in cryo and that it was undergoing conformational changes. Then a few years later, I published some 3D structure, low resolution 3D structure, again showing about the dynamic properties of the change of conformation of this complex during the ATPase cycle. And then this work was quite seminal at the time because it was the first member of a large class of protein, the AAA ATPase, to be studied by cryo-EM. A couple of years later, Baron de la Barre published the first crystal structure of VCP, and it confirmed our cryo-EM structure. It showed that P97 assembled as a homohexamer, forming two stack rings. So if we look at the structure of a monomer, we have an N domain, a D1 domain, and a D2 domain. And basically, 6 D1 domain comes together to form a ring, and 6 D2 domain comes together to form a second ring below the top, what we call the top ring, the D1 ring. And then the N domain is sitting surrounding the D1 ring. There are nowadays more than 200 structures of VCP, full length and domain deposited in the PDB, so I won't talk about all of them, but I'll just mention a few today. And so about the function of P97 or VCP. So P97, first of all, is one of the most abundant protein in the cell cytoplasms, and it has been shown to be a master regulator of proteinostasis. It's been pretty much involved in nearly all cellular function, it being from chromatin-associated degradation, associated degradation of the ERAD pathway, autophagy, Golgi assembly, mitochondria-associated degradation, ribosome-associated degradation. So how does it perform all these functions as a key regulator? And the multitude function has been associated with the property of VCP to unfold proteins and disassemble protein complexes, it being protein or protein DNA complexes. And some of these dissociated proteins, but not all, are then degraded by the proteasome. And some of the recognition of these proteins that are being dissociated or degraded are recognized thanks to their ubiquitin signature, but again, not all. And so as an overall, cellular homeostasis and proteostasis, so cellular homeostasis sustain optimal condition for cell function, energy balance, and stress adaptation. And so in a way, like VCP is involved in regulating protein unfolding and degradation, and these are essential to maintain cellular homeostasis. So on this for two reasons. First, because protein deteriorate over time, and as such, they must be discarded or recycled, else they form toxic aggregate in the cells that quickly become toxic. And the second reason is that many cellular processes depend on protein complex disassembly and protein degradation, and this including cell cycle regulation, signaling pathway, membrane fusion, and DNA function. Also, not surprisingly, mutation in this protein have been reported to have a dire pathological consequence. And Virginia Kimoni was the first one to report in 2008 that mutation in VCP resulted in a disease in patients where patients had accumulation of protein aggregates in the cell cytoplasm on bone and brain. She then showed that these aggregates are also found in muscle. And so currently there are many mutations in VCP have been found, creating a rare disease with no cure, which have mostly late onset. So patients tend to be over 40 when they start developing the disease. So these are, the signature of this disease is protein aggregate in bone, brain, and muscle. However, very recently there was a report from Parkville that showed that, where he found 13 young patients that also seemed to have mutation in their VCP protein and were developing neurological disorder. There currently are about 16 mutations, all single amino acid mutation in the protein. And what's characteristic is that most of those mutations are found at the interface between the N and D1 domains. So in my lab, we became quite interested in understanding how these disease variants impact the conformation of VCP. And so Dalia, who was a PhD student I co-supervised with Martin Latterich at McGill University, showed that surprisingly, this mutation did not impair the oligomerization state of VCP. So the protein still form hexamer in all conditions. But she also showed that this mutation actually resulted in an increased ATPase activity of a protein. So it's very rare to find mutation that actually increase the activity of an enzyme rather than decrease it. But this is the case for VCP. And then she showed using fluorescence, mostly looking at the fluorescence of a tryptophan 472 in the D1 and D2 linker, and using proteolysis, that even though the mutations were between the N and D1 domain, it seems that they affected the conformation of the bottom ring, so the D2 domain. This work was followed by Dries Montassi for Dries Lajoie in my lab at McGill, and he showed that among the mutations, mutation R155P was most likely to be most different from the other, will show the largest conformational change from the other mutation, and this using isoelectric focusing gel. And then he initiated some low resolution cryo-EM, so those were all CCD data, where he compared the cryo-EM structures of VCP wild type and mutant in the absence of nucleotide. He showed that in both structures, the N domain, in the absence of nucleotide, was coplanar to the D1 ring, And then he also showed that there seemed to be a different conformation in the D2 domain. Do you see my mouse when I talk? I wonder if I do that. Oh, that would be better. And then Dries also showed that this mutation seems to impair the interaction of VCP with a set of proteins that regulate its function, the cofactors that I will discuss a little bit later. Then Dries collected a large set of data, again on the CCD at the time, where he looked at the mutant protein on the wild-type protein, on this in different nucleotide conditions. So ATP gamma S to mimic ATP, the ATP state, ADP, and the absence of nucleotide. What he saw is, using multiple classification, that the protein was quite flexible, and that only in the presence of only the wild type, in the presence of ATP gamma S, adopted the different conformation as shown here, where the end domain is seen above the D1 ring. This work was followed by CPD Valimer here at my lab at University of Melbourne, where she calculated a high resolution of both the mutant protein and the wild-type protein in the presence of ATP gamma S. So she saw several things. So her studies confirm that the end domain was located above the D1 ring for the mutant, or the disease mutant, in the presence of ATP gamma S, but the end domain was seen coplanar to the D1 ring for the wild-type protein. But however, in this reconstruction, you could see that the end domains were not equal to the rest of the density, and they were poorly defined in both the mutant protein and the wild-type protein. And so she worked really hard trying to solve this conformational heterogeneity. So she used symmetry expansion on extensive masking and a lot of things. But no matter what type of discrete classification she used, she could not really resolve the end domain in all the protein. So instead, what she got is she got classes where she could see the end domain fairly well, and then classes where the end domain were pretty much totally absent. And so in the case of a wild-type protein, she was able to see the end domain for about 38% of the monomer within all the hexamers. She did the same analysis for the wild-type protein. This was a little bit not as bad, where she could see the end domain for about 57% of the particles, and the others, the end domain, were completely washed out. Nonetheless, using the 38% of the protein and the 57% of the protein, she calculated a very good reconstruction of the mutant on the wild type, and then she started analyzing the difference. So what she saw, that despite different position of the end domain, the two structures were very similar to one another. She observed ATP gamma S in all the nucleotide binding pocket, but she only saw a tiny, tiny difference in the nucleotide binding pocket for phenylalanine 360, which adopted a different conformation, so flipped away from the nucleotide. She then she also saw other differences between the two structures, mostly in the position of the linker between the N on D1 domain and the linker between the D1 and D2 domain. And also, so this suggested that the mutation disrupt regulation of VCP domain. So you could imagine that you have a hexamer composed of for a total, so three domains per monomer times 6, 18. So the coordination of this domain is very important for function. And she saw that the mutant really affected the coordination by changing the conformation of a linker. She also showed that there was a small domain movement in the D1 ring, especially in the alpha subdomain, which is important for coordinating the motion between the D1 ring. So overall, our work showed that mutation disrupted the allosteric regulation of VCP. But then, looking back at the data, you have to remember that these high-resolution structures were only obtained with 30% of a particle in one case or 57% of a particle in one case. And this was quite bothersome because it obviously shows that we're not capturing the whole range of conformation adopted by the protein. And this is contradictory of what we know for cryo-EM. So in principle, because we have our protein in multiple orientation in the ice, in absence of crystal, cryo-EM data should be able to capture all the conformation of the protein complex. However, when we have different conformation, this leads to blurring of a 3D reconstruction, or in some extreme cases, in some densities being completely absent. So in this case, we're talking about 630 kilodalton domains being absent in most of our reconstruction. This is a lot. And then, so CPD carefully checked that she didn't have any proteolysis, and that she was working with a full-length protein. And then she turned in working with Ashish Zeti, a postdoc in our lab, to try to understand why she was not seeing this end domain. And so she first turned with Ashish and Swati to cross-linking mass spectrometry. So I'll explain cross-linking, the methodology of cross-linking mass spectrometry. So you get your protein in solution, then you incubate it with a linker. So she used the SSO. So this is a linker about 10.3 angstrom. And then so you incubate the linker, cross-link whatever residues or lysine residues that are in close proximity in the 3D structure, and then you stop the reaction and you do an extensive trypsin digestion or proteolytic digestion, getting small peptide that you then resolve by mass spectrometry, and this technique allows you to determine cross-link peptides, so a residue that were in close proximity in the 3D structure. And so using this method, Ashish and ZPD showed that some of the crosslinks were explained easily or observed were explained based on the structure, as the residues were within 25 angstrom of one another in the 3D structure. However, they also observed a crosslink between residues that are very far apart from one another, so higher than 25 angstrom, some are really higher than 35 angstrom in the 3D structure. That indicates to us that there are some flexibility in the data set that cannot be explained by cryo-EM structure at the moment. And so what she shows, like what you can also do with cross-linking data is plot residues that are interacting with one another on the 3D structure. So she plotted this on both the upward conformation on the complanar conformation, and showed that some really interacting residues were far apart in the structure. And so she did the work, and then she could also compare the wild-type protein and the mutant, and showed that the dynamic of those two properties were different, as a set of cross-linkers were unique for the wild-type protein and some unique for the mutant. Then she also carefully repeated the work with in the absence of nucleotide, in the presence of ATP gamma-S, and in the presence of ADP to capture the full range of motion. And she showed that both wild type and the mutant change conformation during the ATPase cycle, which is not surprising, but also that the conformational dynamic of both the wild type and the mutants were different from each other and in any nucleotide condition. So then, this is great. It shows that we have conformational heterogeneity, which we suspected because we could not see all the density in our 3D EM map. But somehow it was not fully satisfying because we're not able to understand the range of conformation. So to do that, we turn to a collaboration with Slavica Jonik, with a co-supervised PhD student, Rémi Vouillemot. And Slavica was developing methods or image analysis methods to understand or using molecular dynamics to understand conformational heterogeneity, and this from raw cryo-EM data. So basically the movie or the images collected in the transmission electron microscope. And so then I will summarize the problem shortly and explain the difference between discrete and continuous variability. So as mentioned in cryo-EM, on your EM grid, one is expected to have all the possible conformation of a protein. But with different conformations are discrete, so where we have a different set of conformation, then discrete classification, where you split the data in a set number of groups, and being in this case 3, but it could be whatever you do. So you typically try different values and see what works. Then you can get fairly decent reconstruction. However, if you have continuous variability, so where a domain or there's not really a defined set of conformation, then discrete classification perform very poorly, and no matter how hard you try, if you try to group the particle in defined set of particles, you always end up with blurry reconstruction. Maybe a stable core, but then really big density or even missing density for the rest of the protein. And this is exactly what CPD was getting. So here, if we look at the reconstruction she obtained for the mutant in the presence of ATP gamma-S, applying C1 symmetry, or basically no symmetry, then what she could see is that maybe, she could see a bit of density for 2 of the domain, and no matter if she changed the thresholds, some kind of end domain became visible, but one end domain was never ever visible. And so then I explained to you, she did fancy expansion symmetry and masking, she was only able to resolve density for the end domain in 38% of the particle. Obviously, applying C6 symmetry doesn't really help in resolving those domains, as the densities becomes really, really blurred. And so Slavica was developing a method to understand conformational flexibility using molecular dynamics. So I'll do a quick overview of molecular dynamics on how we can use molecular dynamics and normal mode to understand conformational flexibility. So typically, if you use standard molecular dynamics alone, it's really not an effective method to understand conformational changes, as molecular dynamics struggle to overcome high energy barrier. However, if you use bias potential, so for example, a cryo-EM data, it being a 2D or 3D volume, then you can overcome this limitation of a molecular dynamic, and then you can reach your target. However, this using molecular dynamic, even biased molecular dynamic alone, is highly computational expensive. It takes a lot of time, and this is because there's the small step size of molecular dynamics and the large number of degrees of freedom. This can somewhat be reduced using coarse grain, but is still very low and expensive. So alternatively, what people have been using is normal mode. And so normal mode just reflects the large motion possible for a protein, so it tends to be fast. But unfortunately, normal mode simulation tend to be not realistic, and the structure tend to be deformed. So then what Remy showed is that if you're able to combine normal mode analysis for the large conformational changes and combine it with molecular dynamics, you can reach the target a lot faster. And this is what Remy applied to his structure. So basically, in this method, you need to start with one atomic structure and then a set of raw data, so 2D EM images. And then you take the 3D structure and then you deform it so that it matches the density seen in the 2D projection. So the output of this method is a large series of atomic models, and that you can then classify to understand the conformational heterogeneity. And you use principal component analysis such as UMAP, that works really well for this method, to plot the different structure in a vectorial space. So you have component 1 and component 2, and then each little dot in this map corresponds to one model. So as it becomes for 200,000 particles becomes or 200,000 models too difficult to see, we color by a heat map. So blue indicates a region of a map where we have a lot more model, and red less model. So in short, what we could see here is that we had two major clusters of conformational conformation, one a large cluster shown here and a small cluster. And so the large cluster did not correspond to one conformation, as if we were to put the model together and look at the RMSF, we could see a lot of variability on the end domain greater than 10 angstrom. Actually, what we were able to do, if you take each model individually, you can see which conformation it sits in. And then what we saw is that we had domains where all the end domains were more or less coplanar, one up, then one down, one further up, and so on and so on. So a lot of variability in the data. And in order to understand this variability with greater detail, we then split our hexamer into monomer again, and then analyze the conformation of each monomer. And so that we saw that indeed we had continuous motion of the end domain, where in the middle position that we referred as 0 degree correspond to the end-up conformation that CPD resolved using high resolution, but that indeed we have a large variability or large motion of this domain, where the end domain rotated from minus 30 degrees to plus 30 degrees in respect to the central position. And then with this data, we could then understand if the motion of the end domain was coordinated in some ways. And we split this map into 3 categories: anything smaller than 20, anything from minus 20 to plus 20, and plus 20 degrees or higher. And then we could analyze or get a histogram of a different conformation. So basically what this histogram is telling us is that all possible motion of the end domain is possible, except for this class here, where the end domain will be adjacent, all the adjacent end domain will be in a totally different conformation. So basically the data says that yes, the end domain is flexible, but we have at minimum 2 end domains in a very similar conformation. And then also, quite strikingly, what this analysis showed us is that we had a very small class of conformation, and this corresponded to only 2% of the particles. So this demonstrates the power of the technique, as we were able to determine the conformation of a very small subset of particles. And then in this conformation, what we saw is that the D1 and D2 rings were in an open conformation. And so then this was interesting. And then actually it was also more interesting because this open conformation corresponding to a substrate engaged conformation. So since the original studies from Baron de la Barre, which showed a coplanar conformation, there were a few high-resolution structures using cryo-EM, and most interestingly, recently there were some conformation that captured VCP in the action of unfolding, VCP and the yeast homologue CDC48. And so basically what this structure shows is that the compact structure was no longer compact, it was opening and the 2 rings were split. And this led to models such as these for mechanisms of unfolding, where VCP would thread a protein through its central pore, unfolding it 2 amino acids at a time in what we call the hand-over-hand mechanism. But then you could go again, okay, so you got this conformation. This suggests that the mutant protein is a little bit more active than the wild type, and it spontaneously adopt, or a small subset of protein spontaneously adopt the substrate engaged conformation. But is that real? You know, it's molecular dynamics. People are always a little bit skeptic about that. So to confirm this, we use hydrogen exchange mass spectrometry to look for these different conformation. So in hydrogen deuterium exchange mass spectrometry, the principle is that residues that are at the surface of a protein will exchange deuterium a lot faster than embedded residues in the protein. So in principle, in practice, you get your purified protein, you incubate it with a deuterium, and then you incubate for a set amount of time, allowing this exchange to happen, and then you do pepsin digestion, run mass spec, and detect peptides that have uptaken the deuterium. And then analyze the data, either in linear form or in 3D. So then what you can do is then plot this region of in the protein that are exchanging deuterium, or that are uptaking deuterium very quickly. And so what we saw is that this region of the protein uptook deuterium much faster than others, and quite surprisingly, we saw that the deuterium uptake was mostly in the D2 domain, so this domain that is opening in our mutant. What we saw also is you can then compare the uptake of a wild-type protein with a mutant protein, and we saw two things. So one, a peak right at the N-terminal peptide shown here. And this N-terminal peptide, or this change in the N-terminal peptide confirms that we are, or is consistent with the upward conformation of the N domain, as in the coplanar conformation, whose residues do not bind to the D1 domain, so are free, and in the upward conformation, they bind tightly to the N domain. So this suggests that, or confirms that, we are capturing the N conformation in our cryo-EM data. And then the other type of difference, we saw some conformation or some differences in the D2 domain. So this is the domain that is consistent with opening of the D2 domain, as shown here. Okay, so I'll shift gear a little bit now. And so why is VCP so interesting? It's a rare disease, but it also has been identified as a therapeutic target, and this for cancer treatment, where first of all VCP was shown to be at elevated level in cancer cells, on all the way to showing that inhibiting VCP in xenograft models was relevant for treating cancer. Recently also, VCP was shown as to be important for viral replication, and we also suggest that VCP may be a good target to treat parasitic disease and mycobacterial infection. Also, just to summarize a little bit, what we have is, in the absence of substrate, VCP form a very compact structure resembling two stacked rings on top of one another. But then in the substrate, or in the action of unfolding protein, the two stacked rings are adopting a split washer, so they're broken, they separate from one another, and they also, they're no longer forming a rings, but a split washer. So what we believe is that cofactors are very important in for this change of conformation and for recruiting substrate to VCP, as well as for loading the substrate to the protein. And indeed, many cofactors, so over 20 cofactors, have been found to associate with VCP, and some of them are very specific for a specific function of VCP. And so several people in my lab have started work on understanding how cofactors recruit and engage substrate and promote its unfolding. We are using combined combined structural methods, including single particle cryo-EM, cellular and biochemical assay, cross-linking, mass spectrometry, HD-XMS, and NMR spectroscopy. I won't have time to present the work on cofactors today, as it's time. Hopefully I'll be able to show that to you at some other stage. But just tell you that's very important. So just imaging VCP unfolding protein, and if unfolding does not happen in a regulated fashion, you will get a real mess very quickly. Okay, so then finally to conclude, I'd like to acknowledge people in my lab who've done the work. Most of them I've mentioned during the talks: Remy, Messam, CPD, Gigi, Bag, George, and Ashish and Masan, who work on the VCP project, as well as all the other members who are working on different projects. And I would also like to thank the Bio21 facilities that are essential for our work within the Ian Holm Imaging Center for cryo-EM, but also the mass spec facility and the protein facility. On this, I'll end my talk and I'm ready to take questions.
Thank you, Isabelle. It was a great talk. Now the floor is open for questions. You can raise your hand and we can go one by one, or maybe put it in the chat box and I can read that for you. Oh, maybe I can start. I haven't done any MD, and it was great seeing that. I just wanted to ask, what model do you start with in MD? Like, what was the starting point? What model did you take for your MD?
What module?
Yeah, so where did you start this?
So we start with the structure on
the structure. Oh, yeah. I mean, in general, I just don't know about. And is it the same model for the biased one and the normal one? And just like.
Yes, but then in the bias one, you basically add another function in your MD simulation, and this function is your 2D image.
Oh, okay. Cool. The map you were saying?
No, each image. So.
Oh, okay. The micrographs.
Yes, the micrograph. Well, actually, kind of not really the micrograph, the box particle, your box 2D image.
Okay. And what software?
So you use this image as a bias in your MD simulation.
Okay. Anyway, we have a question. I do not know the name. It's TNet. Do you want to unmute yourself and ask a question? Oh, it's Theo.
It's me. Yeah. Hi. Thanks for the interesting talk, Isabelle. I was also curious about this MD cryo combination software you've got, and particularly its applicability to the continuous heterogeneity and pulling out smaller subsets from your data. I was wondering if you compared this strategy to other available algorithms like CryoDragon or maybe 3D VMA, and how it works to those.
Okay, this is a great question. Thank you for asking. However, first of all, I'd like to mention that MD Space is distributed within the Scipion platform if anybody wants to try it. But then I would also like to mention that the principle of this method is different from other methods available, where it's unique by the fact that we are fitting the data directly to the 2D image. while those other methods that you mentioned kind of do almost discrete classification or get a large range of different individual volume, and then they use MD to fit into the 3D volume. What we're doing is different because we are fitting to the 2D image. And also for some of you who may be interested, how fitting is not limited to 2D image, as we showed using in another software using MD Tomo, that we can then look at each individual subtomogram and do the fitting within each subtomogram. So without averaging different volume together. So this, because now we're only looking at densities for one particle at a time, it being in a 2D projections, or in a subtomogram little box, then we can really understand the conformational heterogeneity, kind of really describe all the possible conformation adopted by a protein.
We have another question in the chat box. Maybe I'll just read that. It's from Vivek. It says, lovely talk. I was wondering how you think the R155 mutation is altering the N-terminal residue positioning and several other chains.
Yeah, so I don't, unfortunately, I don't think the answer is very simple. So we know that, which slide should I show there? Okay, so let me share this one here. So we know that all the mutations are located at basically in this region, so at the interface between the N and D1 domain. And so it seems that almost together they make some kind of sticky regions, and having mutations there free up this domain or remove some of the collective binding or interaction between the N and D1 domain. What we were surprised is we didn't see like a large conformational change when we did in this domain, when we did in the mutation. So it really is to be kind of subtle and impacting the interaction between the two domains.
I think that's it. Maybe I can ask one more. What software did you use for the MD you did?
This is GROMAX. So basic. Well, no, actually, no. Sorry. What we did is we use Genesis, and Genesis modules have been incorporated within the software MD Space that I mentioned.
Okay.
So Genesis was the one used. So it's the software developed by Florence Tama in Japan.
Okay. Cool. I think right on time. I guess we don't have any other questions in the chat. And yeah, thank you, Isabelle.
So thank you, everyone.
Just before leaving, I want to address that the next seminar will be on March 11th. And the next speaker is Professor Isabelle from Beihai, and we hope to see you there. And thank you so much, everybody, for joining. Thank you, Isabelle.
Take care. Bye.